Hello. The next subsection talks about high resolution expression mapping. Basically, as I mentioned previously, low resolution means you're looking at the expression of one gene at a time. But if you're talking about high resolution expression, you're talking about looking at the expression of multiple genes or even the whole genome. The other thing you can also do is to map where the gene is being expressed. So how do you do that? You actually, well, you design as such that the product or the RNA product, when it gets expressed, carries either it carries a dye or either it carries a sequence that allows you to stain it. And this example, if you look at B, what it says in B is that in situ hybridization. The example shows expression of the FGF8 fibroblast growth factor gene in a chick embryo at Hamburger Hamilton stage 20, right? Of embryonic development, transcripts were labeled with an antisense deoxygenin labeled FGFJ. So what they have is that they know this gene is being expressed, and they design a probe that will bind to it, and this probe carries a substance that allows when it's reacted, it gives light, it gives color so that you can actually see it. Okay. I mean the following sections following on that tells you about how they um, study. You can use antibodies. Why are we using antibodies? Well, antibodies, what they do is it's very specific. It can be used to detect certain types of proteins. If you do probes and you know whatnot, you're actually talking about RNA. But if you want to measure proteins, you use antibodies. You raise antibodies. Antibodies can be very specific. So similarly, if you design antibodies for a certain protein, then you know, and this antibody is designed to carry some sort of a reporter molecule. Reporter means something that gives light, right? So the higher concentration of the antibodies, that means the higher expression of the gene that you're looking for. Right? So it's direct correlation. Polyclonal and monoclonal, whether it's a mix or whether it's only one type. And then 7.2 talks about high throughput gene expression analysis. This is what people are doing nowadays. They use NGS, next generation sequencing, where it allows you to sequence the whole genome, or even the whole RNA, which is called the transcriptome. Or you have also methods like MALDI-TOF, mass spectrometry, where you actually look at the full complement of the proteome. So it depends on what type of. I mean, there are certain labs that are very strong in transcriptome analysis, whereas other labs are stronger in looking at the protein. So you know, so normally when you have papers, one lab, they did not seldom you see certain lab that does both transcriptome and proteome unless you're a very rich lab. But normally, in a lab that you do on your own, you either do the transcriptome analysis or you do a proteome or you collaborate. So transcriptome means looking at all the expressed sequences, and then what you get is a transcriptome profile. There are various platforms. The most popular is the Illumina system, the HiSeq, the MySeq, and so these are the machine that does the sequencing. Later on, when we talk about, again, in my series of videos in the YouTube under the playlist Human Genetics, there are several videos that talks about different technologies for sequencing. Right, have a look at that as well. Microarrays. No, I don't think I'm gonna go through this one by one, because I think they are just different methods. maybe I can just explain. This is before we do sequencing normally. At that time, there are a lot of work that does this microarray. What they do is this. For example, you have let's just assume that you have two types of cells, normal cell and a cancer cell, right? A is a normal cell, B is a cancerous cell. So you extract RNA from A, you want to see what's expressed in the normal cell, and then you extract RNA from B and you want to see what's expressed in the cancer cell. So A, you label it one color. In this case, let's say you label it with red. Normally red is what? Can't remember anymore. I think it's written somewhere there, but never mind. So it says one is red, one is green. So you put this. So now you have a chip or a slide that has the human genes, right? So from the normal cells, you extract RNA and then you let it hybridize. Okay. So basically, what's hybridizing will be if it doesn't hybridize, then you wash it out. So what's left are DNA or RNA that hybridizes what is expressed, and they are colored in red. Okay. Then you wash this off, and then you repeat the same procedure, hybridization with the one in green, right? But before that, you make sure when you did the red, you take a picture. Once you put in the green again, similarly, whatever is present will bind, okay? And if there's no match, can be washed off. So you can see the picture here. You will see that certain wells are very light. That means there's no binding. Certain wells are like red. So this is the express gene. Similarly, you may not be able to see it here. Normally, you require the help of a computer. So the lighter ones means that there's no expression. The darker greens are the ones that with higher expression, right? Then you superimpose, right? Superimpose the green image and the red, and then the red image and the green image. If it is of equal intensity, in the red is intense, in the green is also intense, so red plus green, what you'll get is yellow. If after overlapping these two sequences you only see red, this signal means that this is expressed in the normal cells but it is not expressed in the cancer cell. If you see a signal that is green, that means to say this is the gene that is expressed in the cancer cells and this is not the gene that expresses in the normal cell. So you get three situations, right? Normal specific, red signal. Cancer specific, green signal. Both are expressed equally in normal cell as well as in cancer cells, you get yellow signal. Well, of course, it's both if it's not expressed, then you get a black signal. Okay, so this is where you get Z, which is a combination of red and green. You get X, which is expressed in the red, and then you get Y, which is expressed in the. So similar principles here, but it's just different system. This one is by Affymetrix, and then you get this data, what expressed in X sample, what is expressed in Y sample, and what is expressed in the Z sample, and then you do analysis. Okay, this is by RNA-seq again. These are just different platforms where you sequence all the RNA. Some notes on how this is done. Interesting. This is mass spectrometry. This is for groups that study proteins. When I had my students doing PhD, one of my students, Nadia, was doing transcriptome. Another student is your lecturer here, Dr. Sharifah. She did proteome, where she studied protein analysis. Right, she used a lot of this mass spectrometry, and no, she did DIG. Can't remember what it is, but basically her work is based on protein expression. So we have in my lab at that time we have the RNA group and also the protein group, and sometimes they fight to say that their method is better. So anyway, yeah, I will not go through them in a very detailed fashion, but suffice to say that what they're interested in is looking at gene expression. The latest development, the latest technology is what they call as single cell genomics or single cell expression studies, where the usually you have tissues, the breast cancer tissues, there's normal sometimes you have problems where the tissues are not expressing genes equally, there's a lot of variation and so on, but nowadays you can actually study single cells. You can get one cancer or treated cell, you get one normal cell, and then you can compare. Right, this is the extent of technology nowadays um, that allows you to look at expression of single cells. Um, well, of course, the challenges are understanding cell-to-cell variation, because expression of genes is very dynamic. Meaning to say, one gene and the side and another's, and I'm sorry, one cell and its neighbor may not have similar gene expression. So so this is what it's saying. Yeah, while you have the technology now to just um, sequence or analyze one single cell, but there are variations, and so you need to understand that as well. Okay, I'll just skip through all this. When I say I'll skip through all this, that means you can skip through it as well. I will probably not ask it. I will probably not ask. Will I probably ask? No, I don't think so. I will not. I'm not that bad. Okay. Isolating single cells, how you do this kind of work. Now let's just jump towards the end. Okay, there's a summary page. I think for the summary page, I will do another video. Okay, see you in the summary page. Bye-bye.